today's video lecture, and today's video lecture I'll be solving previous year's NEET questions and discussing the answers. And this is for the chapter, as you can see on your screen, Chemical Coordination and Integration. We are in part 3 of the series. So you haven't watched part 1 and 2, so please do so. Okay, so let's begin. Okay, so question number 1. Adrenaline directly affects on, okay, and then we have this question appeared in 2002 and we have 4 options. A. SA node, B. Beta cells of Langerhans, C. Dorsal root of spinal nerve, D. Epithelial cells of stomach. Okay, so adrenaline directly affects on A. SA node. We'll see how. So the answer is A. Let's go to the discussion. So, epinephrine or adrenaline has a stronger effect on beta adrenergic receptors than norepinephrine. Now, we already discussed this in the previous part. Remember when we discussed the differences between epinephrine and norepinephrine. So, epinephrine or adrenaline has a stronger effect in stimulating the beta receptors. Okay, there are alpha receptors and beta receptors, so we are specifically, don't worry about this in terms of NEET, but just understand there are alpha receptors and beta receptors. Okay, so epinephrine has a stronger effect than norepinephrine, right? And this is one of the main differences between the two. Now what it does, now these beta receptors are present in the heart also. Okay, and they have 2 types of functions. So when adrenaline stimulates these beta receptors, so they act on the heart and increase cardiac output by cardio acceleration. Okay, so the cardiac output is increased and there is increased myocardial strength. So these are the 2 ways by which epinephrine has an effect on the heart. So obviously SA node is being stimulated, right? So heart rate is increasing. So SA node, as you know, are the pacemakers of the heart, okay? The pacemaker cells. So the heart rate is increasing. The number of heartbeats are increasing. So cardiac output would also increase. Along with that, there is increased myocardial strength also. So the heart cells are also working faster, more stimulated. So here you have a table with alpha receptors and beta receptors. If you watch part 2, you'll be familiar with this table over here. What I wanted to show you, yeah, see, so cardio acceleration and increased myocardial strength. These are the 2 functions with respect to heart. Okay, and so epinephrine acts on these beta receptors and they have a stronger effect than norepinephrine. Okay, so to go back to your question, adrenaline directly affects on SA node. Okay, clear? We'll go back to the discussion and then we go to the next question. Okay, so question number 2. When both ovaries are removed from rat, then which hormone is decreased in blood? This again appeared in 2002. And you have your 4 options. You have A. Oxytocin, B. Prolactin, C. Estrogen, and D. Gonadotropin releasing factor. So now the question says when both ovaries are removed. Okay, so clearly C. Estrogen hormone will be decreased in blood. Okay, so estrogen ovaries are the main source of estrogen. Okay, estrogen is secreted from the ovaries. So if that is gone, estrogen hormone will be decreased in blood. So your answer is C. So estrogen is produced by the ovaries. Now, during the follicular phase of the monthly menstrual cycle, now I am showing how estrogen is produced with respect to the menstrual cycle. So if you're not familiar with the menstrual cycle, please go back and read reproductive system and then come back to this question, because there'll be a lot of terms used with respect to the menstrual cycle. Now, during the follicular phase of the menstrual cycle, now follicular phase is a phase which is dominated by estrogen. Okay, there's less progesterone, but there's lots of estrogen produced during the follicular phase of the menstrual cycle. progesterone and androgens, okay, convert to estrogen. Okay, so during the follicular phase, what happens is there is lots of androgens produced, okay, with the help of LH, and there's also some progesterone also produced, okay, and they convert to estrogen. So all of androstenedione, remember I discussed androstenedione before. So androstenedione and some amount of estrogen convert to, progesterone convert to estrogen by diffusing from theca cells to granulosa cells. Now how does that happen? Okay, now theca cells is the place where androstenedione and progesterone is also present. Okay, it moves from the granulosa cells, progesterone moves from the granulosa cells to the theca cells, and androstenedione is produced in the theca cells with the help of LH. Now we'll see that how. Now LH contributes to the production of androgens in theca cells. Now, granulosa cells contain an enzyme called aromatase that performs the conversion and is stimulated by FSH. So, now what happens is, remember that there are two types of cells in that follicle. Okay, there's theca cells and there's granulosa cells. Okay, now say androstenedione or other androgens, androstenedione, I'm just writing andro, is produced in the theca cell. There is some progesterone also in the granulosa cells which move into the theca cells. So the source of estrogen is androstenedione as well as progesterone. The precursor of progesterone is pregnenolone, but we will not go into the chemical reactions or how it is synthesized. It's not for NEET, so much for NEET, but just to help you understand. Okay, so there are pregnenolone is first produced, then it forms progesterone. It also forms then DHEA, which is the precursor for androstenedione, and also progesterone gets converted to androstenedione directly also. So there are a lot of reactions that are happening, okay, which eventually forms testosterone, then forms estrogen. So let's just understand that there are two types of cells, theca cells and the granulosa cells in the follicle. Okay, so androstenedione is present in the theca cells. Then granulosa cells have also progesterone in it, which move into the theca cells, and both of them then allow this conversion to happen to estrogen. So what happens is the theca cells don't have the ability to convert it into estrogen. Okay, they don't have the ability to convert to estrogen. So what happens is they move into the granulosa cells. Okay, see, some amount of progesterone is needed to convert to androstenedione, so that is also that also moves in the theca cells. Now when the final androstenedione is produced, okay, it moves into the granulosa cells. Okay, now this androstenedione is produced with the help of LH. Now this androstenedione moves into the granulosa cells. Theca cells don't have the ability to produce estrogen. It is the granulosa cell which are actually producing the estrogen. But where is the precursor coming from? Coming from theca cells. Okay, that is with the help of androgens, androstenedione specifically. Okay, so androstenedione with the help of progesterone also it moves into the granulosa cells. Now once it moves into the granulosa cells, there is an enzyme called aromatase. This, well, I don't know if they want to up the level of difficulty in NEET, they might ask you this question of which hormone is responsible for forming estrogen. But it is very unlikely. It is it is only when they want to level up the game in NEET in the exam. So aromatase is an enzyme present in the granulosa cells. Okay, now what happens is with the help of FSH, what happens is these androstenediones are converted to estrogen, and this reaction happens in the granulosa cells. Okay, this reaction happens in the granulosa cells. So granulosa contains an enzyme called aromatase that perform the conversion and is stimulated by FSH. So when FSH attaches, there's receptors present there. So when FSH attaches itself, it allows for aromatase to get activated and then converts androstenedione into estrogen. Okay, so progesterone to a much lesser extent was from the follicular phase. Progesterone is not very dominant. It is mostly androstenedione and the androgens which participate, but to certain extent progesterone also helps in forming these androstenediones. Okay, and then androstenediones move into the granulosa cells where this conversion is happening, and that is with the help of aromatase. You need to understand where LH and FSH is, but menstrual cycle is just a play of hormones. That is all. Okay, so you need to know in the follicular phase which is dominant, in the secretory or the luteal phase which hormone is dominant, what exactly is happening. Okay, and yeah, to understand this you need to study the menstrual cycle in a little bit of detail. To answer your question, I'm giving you this information. Okay, so how estrogen is produced in the ovaries. Okay, so this is actually what's happening. So granulosa cells which produce estrogen with the help of androstenedione and some of progesterone, which moves from the theca cells to the granulosa cells, where there's an enzyme called aromatase which assists in the conversion, and that is with the help of FSH. Now if they ever ask you how are androgens formed in the theca cell, that is with the help of LH. Okay, that is with the help of LH. This happens in the follicular phase where obviously, like I previously mentioned, it is dominated by estrogen, right? In the luteal phase, it is mostly progesterone. There is some amount of estrogen. Obviously, not all the progesterone is converted to estrogen. Right? Some amount of it is converted to estrogen. So, estrogen levels are lower as compared to progesterone in the blood in the luteal phase. Okay. So yeah, here you have a diagram of a Raphian follicle, a mature follicle. Okay, and here you see the granulosa. That's the pink region over here. And you have the theca interna. Theca interna is where LH attaches to it. Okay, LH attaches to this and forms the androgens. Okay, androstenedione to be specific. Okay, and this then moves into the granulosa cells where the reaction is happening. Okay, so this is where estrogen is produced. Okay, so remember this hormone aromatase. I don't know if they'll ever ask in NEET, but now you know, right? Clear? To answer your question, when both ovaries are removed from the rat, then which hormone decreases the blood? Clearly estrogen. All right, clear? Let's move on. Okay, so question number 3. Mainly which type of hormones control the menstrual cycle in human beings? This appeared in 2002 again. So you have A, FSH, B, LH, C, FSH, LH, estrogen, D, progesterone. Okay, so mainly which type of hormones control the menstrual cycle? So to give you the appropriate answer, yes, it is C. Okay, it's FSH, LH, and estrogen. So your answer is C. It's not only FSH, it's not only LH, as neither is it only progesterone. So the appropriate answer is C here. Okay, so let's go to the discussion. Okay, so FSH, LH, and estrogen mainly control the menstrual cycle. That is to answer your question, right? Now let's see how. Now I will go through every phase in the menstrual cycle and tell you what changes are happening and what hormone is responsible for it. Okay, and before, I would suggest that before you go through this answer, listen to my answer, I would suggest you go back and read menstrual cycle because it will help you understand it better. Okay, so first phase, I'll go through follicular phase. Okay, follicular phase. Now FSH, when it is secreted from the anterior pituitary, causes accelerated growth of 6 to 12 primary follicles. Okay, and there is rapid growth. Okay, so initially there is rapid growth. Okay, eventually forming granulosa cells and theca cells, that is theca interna and externa. Now we know why these theca cells and granulosa cells are important, right? So initially when FSH comes, it causes accelerated growth of the primary follicles, which forms the basic now structure. It's forming the theca cells and the granulosa cells. Now theca interna produces estrogen, whereas externa forms the external capsular layer. Actually, it's not totally theca interna, it is granulosa cells which is converting it, right? So along with that, it is granulosa cells also which participate. Now after the early phase, granulosa cells secrete a follicular fluid that contain estrogen, right? We've already discussed that. So theca cells and then forms the interna and the externa layers, right? So external layers is like just a capsular follicle layer. Now after that early phase, the rapid growth that is happening after that basic structure has been formed, the conversion takes place, right? Estrogen is formed from androstenedione, a little bit of progesterone also. Then that follicular fluid is called estrogen. Okay, it contains estrogen in it. Okay, this is the follicular fluid contains estrogen, and this accumulation of fluid forms antrum. Antrum is like a space inside within the mass of granulosa cell, which is governed by FSH. Now, once the antrum is formed and the follicular fluid is inside and estrogen is also produced, this is followed by accelerated growth forming vesicular follicles. First, there was accelerated growth, it forms a basic structure, then it slows down, antrum is formed, follicular fluid is secreted, estrogen is produced inside. Now, again, it picks up, again, the growth picks up. Okay. Accelerated growth forms the vesicular follicles. Now we'll see how that happens. Now this is due to presence of estrogen secreted on the follicle causes increase in FSH receptors of granulosa cells. Okay, so when that estrogen is produced or secreted into that follicle, it increases the FSH receptors. Okay, now when FSH is secreted from the anterior pituitary gland, it attaches to receptors. That's how it works. When you study cell membrane and study the cell in detail, there are receptors attached to it. When we speak about insulin receptors, we speak about FSH, LH receptors, there are receptors which accept these hormones. Okay, there are FSH receptors that are increased. Okay, so this creates a positive feedback, making the granulosa cells sensitive to FSH. So what it does is, now the estrogen causes an increase in the FSH receptors, which again sends a positive feedback to the FSH. Yeah, you can keep secreting. So there's an increase in FSH and the granulosa becomes more sensitive to FSH. Now FSH in estrogen promotes LH receptors, okay, allowing LH stimulation, creating a more rapid increase in follicular secretion. So FSH and estrogen combined together, they also promote LH receptors, so allowing LH stimulation now also to happen. So FSH is estrogen and now LH is also increasing. Okay, and this in turn increases it allows a more rapid increase in follicular secretion. Okay, so now this is how vesicular follicles are formed. So ovulation should be later. Now increasing estrogen and LH together causes the proliferation of thecal cells and increase their secretion. Okay, so now we know FSH is also increased, estrogen is also increased. Now it allows LH also together, it allows proliferation of more theca cells. Okay, and they increase their secretion. Now in the uterus, stromal and epithelial cells proliferate in the endometrium. Okay. So now we know what is happening in the follicle part, right? But now at the same time there are changes happening in the uterus also. Okay, so there are changes happening in the uterus also along with the follicle part, which is maturing with time. So in the uterus, what happens? In the uterus, the stromal and the epithelial cells, the connective tissue and the epithelial cells, they proliferate in the endometrium. That means they increase in numbers. Okay, they proliferate in the endometrium. And before ovulation, the endometrium thickens due to increasing number of stromal cells. there is growth of endometrial gland, and there is formation of new blood vessels. Okay, so before ovulation, there's endometrium thickness that is increasing, there's increase in number of stromal cells, there is growth of endometrial gland, and there's formation of new blood vessels. Okay, so these are the changes that are happening in the uterus at the same time that all this is happening inside the ovary. So we just not only talking about ovary, but there are changes happening in the uterus too. And the endometrium becomes 3 to 5 mm thick. Okay, so in the follicular phase, the endometrium is 3 to 5 millimeters thick. Okay, so these are the changes that are happening in the follicular phase with respect to FSH, LH, estrogen. Okay, we know that estrogen is predominant in the follicular phase. Okay, so now once that part is done, what happens in ovulation? Now 2 days before ovulation, there is an increase in LH. Okay, there's a surge in LH 2 days before that. Okay, and FSH also increases 2 to 3 fold at the same time. There's a big jump in the LH, but FSH also increases. Now, both FSH and LH cause rapid swelling of follicle before ovulation. Okay, so they cause a rapid swelling. LH acts on the granulosa and the theca cells and converts them to progesterone secreting cells. Okay, so progesterone plays an important role. Progesterone begins to secrete, it increases in the plasma levels, and you see that it plays a very important role in ovulation. So the whole point of ovulation is to release the ovum into the fallopian tube. Okay, so now the ovum is being developed in the follicle, it's ready. Okay, now it's ready to move out, but needs to break away from all the follicular cells that have formed around it, right? So FSH and LH help in doing that. So what LH does is it produces progesterone secreting cells. Now what these progesterone secreting cells do? Now due to progesterone, proteolytic enzymes from lysozymes cause dissolution of the follicular capsular wall and its weakening. Okay, so these progesterone cells, the progesterone is secreted there, and that progesterone helps in forming proteolytic enzymes, okay, which help in breaking that capsular wall, okay, and weakens it. Okay, so now the whole point of ovum is to release, right? So what you have to do, you need something to help it break away from that whole follicle, right? So progesterone helps in producing those proteolytic enzymes from lysozymes, which help in weakening the wall and to break away. Okay, so it ruptures. Basically, that's what's happening. The another thing is that there is rapid growth of blood vessels into the follicle wall and secretion of prostaglandins in the follicular tissue. So first progesterone does is it helps in forming those proteolytic enzymes, okay, from lysozyme. Also, it allows for rapid growth of blood vessels in the follicle wall. Okay, so this is what happens at the time of ovulation. There's a surge in LH. Okay, plasma surge in LH, and FSH is also increases, but not as much as LH. Okay, so both of them then help in forming these as in allows for the granulosa and theca cells to produce progesterone. Okay, they become progesterone secreting cells. Okay, and then this progesterone then helps in forming those proteolytic enzymes. Okay, so before that, you know that this swelling that's happening, right? Both hormones cause rapid swelling before ovulation. So it's preparing the follicle to expel the ovum out. It wants it out but needs to break away. So progesterone helps in producing those proteolytic enzymes. At the same time, it also helps in forming blood vessels into the follicle wall. Okay, so that's how ovulation happens. Okay, so now ovulation has occurred. The ovum breaks away and goes into the fallopian tube. Now the next phase is luteal phase, or it's also called a secretory phase. Now what happens in that? So now after ovulation, LH stimulates the conversion of granulosa and theca cells into lutein cells. We already discussed this in part 2. Okay, so it forms lutein cells. Okay, so there are fat inclusions in it which gives it a yellow appearance. Okay, and this is called luteinization. Okay, now LH plays an important role here. Okay, and the mass of cells is called corpus luteum. Okay, so LH, it helps, it stimulates the conversion of granulosa and theca cells into lutein cells, and this is called luteinization, and the mass of cells is called corpus luteum. Now, granulosa cells secrete progesterone and estrogen, and theca cells secrete androgens, which are converted to estrogen in the granulosa cells due to the enzyme aromatase. Again, the same thing happens there. Remember, I spoke about the follicular phase, how estrogen is secreted. Similar process happens here. Okay, the androgens help in the conversion to estrogen with the help of aromatase. Estrogen to a greater extent and progesterone suppress the secretion of FSH and LH. This eventually causes involution of corpus luteum. Okay, so corpus luteum is a temporary endocrine gland. Okay, we already discussed this before, and so it secretes estrogen and progesterone. Now it secretes estrogen, progesterone. Now estrogen, progesterone is present in high amounts. Now that has the ability to suppress FSH and LH. Okay, till luteal phase is done, FSH and LH tends to be suppressed, so that the follicular phase does not begin again. Okay, it waits for some time. So that's why these high levels of estrogen, progesterone help in the suppression of FSH and LH, and so this eventually causes involution of corpus luteum, which involution means degradation of corpus luteum. Estrogen causes additional cellular proliferation of endometrium, whereas progesterone causes secretory development and swelling of endometrium. So luteal phase, now again in the endometrium, in the uterus, estrogen causes what? The additional, now already in the follicular phase it has caused proliferation of endometrium, but now additional cellular proliferation, now it's just producing more cells and more cells, it's thickening in size. Okay, of the endometrium, whereas progesterone causes secretory development and swelling of endometrium. Now, what are the changes that are happening? Okay, the cytoplasm of stromal cells increases, lipid and glycogen deposit increases, blood supply to endometrium increases. Okay, so what it does is basically luteal phase is a phase where the uterus thinks that if implantation does happen, that means if the ovum is fertilized, so what it does is preparing the female for implantation if fertilization happens. So fertilization is preparing the body for fertilization as well as implantation. So let's say fertilization does happen and eventually implantation does happen. So this zygote that is there, that's produced, that is formed, needs some nutrition, right, to survive. So luteal phase allows that to happen. Okay, it is preparing the body for pregnancy basically. So now corpus luteum will live, involution will not happen if pregnancy does happen, right? But involution will happen if pregnancy does not happen. Okay, but that's a different thing. To understand luteal phase, let's think of it as this way, that the body is preparing the woman for pregnancy, the uterus for pregnancy. Okay, so if that's why all these changes happen, there's cytoplasm of stromal cell increases, lipid and glycogen deposit increases, blood supply to endometrium increases. All these changes that are happening allow for the fertilized ovum to finally survive in the uterus after implantation. Okay, so luteal phase is basically preparing the uterus for pregnancy. Now, thickness of endometrium increases to 5 to 6 mm. Previously, we saw it was 3 to 5 mm. Now it is 5 to 6 mm. Now, we come to the last phase, the menstrual phase. Okay, it is caused by the reduction of estrogen and progesterone. Remember, before estrogen, progesterone are high. So FSH and LH were suppressed. Now slowly towards the end of luteal phase, for before menstrual phase begins, progesterone and estrogen levels drop. Okay, so then that again increases FSH and LH secretion from the anterior pituitary. There is decreased stimulation of endometrial cells and involution of endometrium due to release of prostaglandins. Okay. So now the uterus realizes that there is no pregnancy that's going to happen. Okay, there's no implantation, there's no fertilized ovum, nothing is happening, right? So there is no need for all this, for all this drama to happen. So you might as well just remove all that is formed. So the endometrium lining, all that is formed, all that gets degraded. Sorry. So involution of endometrium happens during that time. Okay, there's decreased stimulation of endometrial cells during that time. So prostaglandins play a very important role. They are vasoconstrictors and they allow for the expulsion of the endometrial lining. Now this initiates necrosis of endometrium, especially of the blood vessels. Remember, there were additional blood vessels that were formed previously, right? Now all that is gone. Now this prostaglandin that is there initiates the necrosis of the endometrium, especially of the blood vessels. That's why you have blood in your menstruation. So within 48 hours, all the superficial layers of endometrium has desquamated. That means this peeling off of all that proliferation of all the stromal cells and all the cells that were endometrial cells that were produced, that were formed there. This mass of desquamation and the effect of prostaglandins allow for uterus to expel the contents from it. So prostaglandins play a very important role. Okay, you feel those contractions, those who go through menstruation, you feel those contractions. Okay, that's like the uterus expelling out all the contents from inside. Now normal menstruation causes loss of 40 ml of blood and 35 ml of serous fluid. Okay, there's a presence of fibrinolysin which causes non-clotting effects, anti-clotting effects in the menstrual fluid. Okay, that's how it allows for blood to move out. It doesn't allow it to clot because of fibrinolysin. Again, this is a potential NEET question if they ever ask. The menstrual phase lasts for 4 to 7 days, and by this time the endometrium has re-epithelialized. Sorry. What happens is, again, the drop of estrogen and progesterone allow for LH and FSH to begin secretion. So again, now it is actually the ovary is going back into the follicular phase again, right? The whole cycle begins again the next month. Let's say if you have a menstrual cycle of about 28 days. So the menstrual cycle lasts for about 4 to 7 days, and by this time the endometrium also slowly as the cells are moving out, there is new epithelial cells forming again to restore that uterus again, the endometrial lining again. Okay. Oh yeah, so this is a diagram, it's actually graph to show the changes in estrogen and progesterone and LH and FSH. So if you like, you can note this down. Okay, and you see over here, like I said, so the follicular phase, which is before ovulation, before this dashed line, the dotted line over here, it is dominant. Estradiol or estrogen is dominant over here, and progesterone is dominant in the luteal phase because of the formation of lutein cells. Okay, estrogen is less compared to that. Okay, so you see also a little increase in progesterone, which helps in the ovulation, right? How progesterone is important in ovulation. And you see this plasma surge of LH. Yeah. And a little bit of increase in FSH, 2 to 3 fold. And together they help in ovulation. Predominantly it is LH. To a certain extent it is FSH. Okay, so I have briefly explained the menstrual cycle to you, but I would suggest you go back and read NCRT for NEET And you can also come back to this video, and if you feel like there's additional information in and you feel like there's additional information in this, you can always note it down, right? So to answer your question, mainly which type of hormones control the menstrual cycle, it is C. The appropriate answer is C. Okay, so question number 4. Which set is similar? This appeared in 2001. A. Corpus luteum graphene follicles. B. sebum sweat. C. Bundle of his pacemaker. D. Vitamin B7 niacin. So which set is similar? It is A. Corpus luteum graphene follicles. Answer is A. Let's go to the discussion. So graphene follicles or mature follicle after ovulation convert to corpus luteum. Sebum is an oily substance produced in the sebaceous glands. Pacemaker is the SA node in the heart, and bundle of his is a part of the electrical system of the heart in conducting nerve impulses. We've already discussed this in the circulatory system, the body fluids and circulation, right? Vitamin B7 is biotin and niacin is vitamin B3. Okay, this was a pretty simple direct question. You had to know that graphene follicle and the relation with corpus luteum. Okay, clear? I guess we can move on to the next question. Okay, so question number 5. Melatonin is secreted by, and this appeared in 2000. A. Pineal gland, B. Skin, C. Pituitary gland, and D. Thyroid. So melatonin is secreted by A. Pineal gland. We've already discussed melatonin also in part 2 in much detail. I'll not go through this in detail over here. Okay, so please go back and watch part 2 of this series, and I've explained melatonin in much detail there. So melatonin is secreted by pineal gland, and the pineal gland is located on the dorsal side of the forebrain. So melatonin plays a very important role in maintaining the circadian rhythm, and I've explained how that sleep-wake cycle is maintained with the help of melatonin. Okay, so you can go watch that part. But to answer your question, yeah, it was quite direct. Melatonin is secreted by pineal gland. There was no doubt in that, right? We'll move on. Okay, question number 6. The gonadotrophic hormones are secreted by, this appeared in 99. A. Anterior lobe of pituitary. B. Interstitial cells of testes. C. Adrenal cortex. D. Posterior part of thyroid. So it is gonadotrophic hormones are secreted by anterior lobe of pituitary. Answer is A. Let's go to the discussion. So gonadotrophic hormones are FSH and LH and secreted by the anterior pituitary gland, right? This again direct question, direct answer, right? You need to know for NEET you need to know which hormone is secreted from which part of the brain, okay, and where it is, which organ does it affect, and what are the functions of that hormone, and if there is hyposecretion or hypersecretion, what are the changes that are happening in the body due to that. Okay, so the pathology part of it. But to answer your question, yeah, gonadotrophic hormones are secreted by the anterior pituitary gland. Okay, next question then. Question number 7. Diabetes is due to, this appeared in 99. A. Enzyme deficiency. B. Iodine deficiency. C. Sodium deficiency. And D. Hormonal deficiency. Now we also discussed diabetes in much detail in the previous parts. So diabetes is due to hormonal deficiency. Yeah, answer is D. Let's go to the discussion. So diabetes is due to deficiency of hormone. Diabetes insipidus is due to deficiency of ADH or vasopressin. Okay, that's another name for ADH. ADH is antidiuretic hormone. It's also called diabetes. So that's why I mentioned that. So that is also due to deficiency of a hormone. And diabetes mellitus type 1 is deficiency of insulin. Type 2 diabetes is different from type 1 diabetes, so don't confuse the two. Okay, now the question says due to deficiency of hormone, diabetes mellitus type 1 is due to deficiency of insulin, and diabetes insipidus is a deficiency of ADH. Okay, so these two are the different diabetes that exist due to deficiency of a hormone. All right. Okay, clear? I guess we've answered this question. Diabetes due to hormonal deficiency. Great. We'll move on to the next question. Okay, question number 8. Calcitonin is a thyroid hormone which, okay, we know it's a thyroid hormone which does what? Okay, this appeared in 1998. A. Elevates calcium level in blood. B. Has no effect on calcium. C. Elevates potassium level in blood. D. Lowers calcium level in blood. So your answer is D. It lowers calcium in blood. We'll see how. So calcitonin is a hormone secreted from the parafollicular cells, that is C cells of thyroid gland. Okay. And calcitonin decreases plasma calcium concentration and is found to have high activity in young animals rather than in adult animals and humans. Now, this is a very important information. Okay, so you see calcitonin decreases plasma calcium concentration, yes, but it is more active in young animals than in adult animals and even in humans. Okay, and when we'll see how that happens. First, let's understand the functions of calcitonin, what it does. Now, calcitonin, what it does is it decreases the osteoclastic activity of the bone, thus allowing for deposition of calcium salts. Okay, so what it does is how it decreases the calcium levels. It will decrease the osteoclastic activity, so it allows for more deposition of calcium salts. Okay, and this process is dominant in young animals because of rapid absorption and deposition of calcium. Second is it decreases the formation of new osteoclasts. Firstly, it decreases the activity of osteoclasts that are already there. Now, that is an immediate effect. What you see, the decrease in osteoclastic activity is immediate. Okay, you will immediately see a change in the calcium levels. For a prolonged effect, what it does is it decreases the formation of new osteoclasts. Okay, so hence over a prolonged period of time, it decreases the number and activity of osteoblasts and osteoclasts. So this happens over a long period of time where new osteoclasts are not being formed, and slowly as time moves on over a period of time, it decreases the number and activity of osteoblasts and osteoclasts. Now, there's a minor effect on kidney tubules by allowing calcium secretion, but it is minor. It is not as strong as how PTH acts on kidneys. So, there's a very minor secretion in the, effect in the kidney tubules where it allows for secretion of calcium. Okay, so the first point where that immediate effect that is there, that is very important. Okay, and it is seen in young animals and also in human children. Okay, where you see what happens in young animals and human as in children, the rate of absorption and deposition of calcium salts is very high as compared to adults. Okay, so there is a tendency of calcium that can increase and it needs to be decreased. Okay, so that is why calcitonin plays a very important role in young animals and in children as compared to adults, because the rate and absorption of calcium salts is high in them as compared to adult humans and adult animals. Yes, so calcitonin has a weak effect in adult humans, like I already explained, and as PTH tends to override the calcitonin effect. Okay, so in adult humans also, PTH has a very dominant effect in the body as compared to calcitonin. Okay, so you will see this calcitonin effect being very strong in children as compared to adults. Okay, like I've already explained, the rate of absorption and deposition of calcium salts is high in children as compared to adults. So it has a very weak effect in adult humans. So in human adults, the daily rate of deposition absorption of calcium is low as compared to children. Hence, calcitonin plays an important role in children. Okay, this is good, trivia to understand where calcitonin has a higher, stronger effect. Okay, so in adults, in humans, what we see is that PTH tends to override. Okay, it is very less likely for calcium to increase in adult humans as compared to children, and we know why. Because the rate of deposition absorption of calcium is low in human adults as compared to children. Okay, so this is a good point to remember. Okay, when you're studying the differences between PTH and calcitonin, how they have an effect on your body. So to answer your question, calcitonin is a thyroid hormone which lowers calcium level in blood. Okay, so this was pretty direct if you knew the answer. Okay, so I guess you can move on to the next question. Question number 9. The hormone which regulates the basal metabolism in our body is secreted from, and this appeared in 98. A. Adrenal cortex, B. Pancreas, C. Pituitary, and D. Thyroid. So the hormone which regulates basal metabolism, that's BMR. Okay, that is D. Thyroid. We'll see how. The answer is D. Let's go to the discussion. So thyroid hormones regulate the BMR in our body. It is based on the metabolic activity which is regulated by the thyroid hormones. It increases the cellular metabolic activity by, so now we know BMR is dependent on the metabolic activity. That's what I was trying to say here. Okay, so thyroid hormones have a direct effect on the metabolic activity of our body. And how that is happening? It increases the cellular metabolic activity. That is how the overall metabolic activity increases because of thyroid hormones, and then in turn the basal metabolic activity rate increases. Now what is BMR? BMR is the metabolic activity of our body when we are at rest. Okay, when we are at rest, that is BMR. Okay, now usually the thyroid hormone has an effect on the normal metabolic activity of our body, and how it does is increases the cellular metabolic activity. What it does is increases the number and activity of mitochondria in the cells. That's how thyroid acts on the cell. First, let's look at it from the cellular point of view. Okay, you have cell, you have thyroid acting on it. Okay, what it does, it increases the number and activity of mitochondria. So what it does is the total membrane surface of mitochondria increases in proportion to the metabolic rate. Okay, so as if you need more metabolic rate, you to increase the metabolic rate, then the number and the total number membrane surface area of the mitochondria increases. Erase this part. Now this increases the production of ATP for cellular energy function. Okay, so as the mitochondria increases, total membrane surface of mitochondria increases, there's more activity happening, then this obviously increases the production of ATP. Now this is one way, okay, by how thyroid increases cellular metabolic activity. The second way is the activity of sodium potassium ATPase enzyme increases with thyroxine. Okay, so this allows for movement of sodium and potassium ions at increased rates. Remember, the sodium potassium ATPase pump is present in lots of areas in our body. You're already familiar with this. So if now thyroxine increases the activity of that. Okay, so now sodium and potassium ions are moving at very increased rates. Now since this process uses energy, it increases heat production. Now, since thyroid controls cellular metabolism, as we have seen, there are two ways by which it controls cellular metabolism. It also causes an increase in BMR, and an increase in hormone quantity causes an increased, causes an increase in, this shouldn't be here, increase in BMR of up to 60 to 100% above normal. Okay, so the thyroid controls cellular metabolism, and so that in turn, overall the metabolism has increased, right? And so that also affects the BMR, okay, the basal metabolic rate also. So if there's an increase activity of thyroid hormones, the BMR also increases 60 to 100% above normal. If there's an increased rate of thyroxine activity happening in the cells and in the sodium potassium ATPase pumps, the whole overall metabolic activity of the body is increased, and that also then increases BMR. Okay, so there is cellular, that's how the flow chart is. Cellular metabolism will lead to full body metabolic rate, and then that also increases BMR. So if this is high, then body metabolism will also increase, and then the BMR will also increase. So if too much activity of the thyroxine, obviously, has, we've already discussed this, thyroxine has a direct effect on cellular metabolism, and that overall increases body metabolism. That overall will increase the BMR by about 60 to 100% of normal rate. Okay, so that's how thyroxine regulates the BMR. Okay. Well, now you know how, because usually they just give you a list of what thyroid does, thyroid hormones do, but now you know in detail how the BMR is increasing. Okay, so if we go back, thyroid, the hormone which regulates the basal metabolism in our body, secreted from thyroid. Okay, so that answers our question. Okay, so question number 10. Which hormone stimulates the secretion of milk from female? This appeared in 1996. So you have A, oxytocin, B, progesterone, C, LH, D, prolactin. So we have to look at the hormone which stimulates the secretion of milk. So stimulates secretion of milk is prolactin. The answer is D. Let's go to the discussion. So now thyrotropin releasing hormone stimulates the release of prolactin from the anterior pituitary gland. Thyrotropin releasing hormone from the hypothalamus, okay, actually stimulates the production of prolactin. Now prolactin inhibiting hormone promotes inhibition of synthesis and secretion of prolactin. Now prolactin is a hormone which helps in the secretion of milk, the production of milk from the mammary glands, right? But now we are not doing that all the time, right? So predominantly the inhibiting hormone is what is stopping the synthesis, and inhibiting the synthesis and secretion of milk, of prolactin, which eventually forms milk. So the concentration of prolactin is usually produced when the female is pregnant. So the concentration of prolactin increases from the 5th week of pregnancy. Okay, so you see a gradual increase in prolactin from the 5th week of pregnancy until the birth of the baby. Okay, when it is 10 to 20 times the normal, as in 20 to 20 times more than the normal non-pregnant female. Okay, so you see such a big high increase in prolactin. Now after parturition, that means after the mother has delivered the baby, there is a loss of estrogen and progesterone, allowing for secretion of milk by prolactin. So that is when secretion of milk is happening, is prolactin is allowing for secretion or production of milk during that time. Now during the whole pregnancy, the gestation period, estrogen and progesterone are produced in high amounts from the placenta. We know that, right? So that is not allowing for secretion of milk. Obviously prolactin is also increasing in the blood, in the body, but that does not allow secretion of milk until parturition has happened. Okay, until parturition has happened, then there'll be a loss of estrogen and progesterone, right? There won't be high amounts of estrogen and progesterone in the blood, and so that is when it allows for secretion of milk by prolactin. Now, prolactin comes to normal levels after birth of the baby. But each time the mother nurses the baby, the nervous signals sent to the hypothalamus cause a surge of prolactin secretion which lasts for 1 hour. So now there is a decrease in prolactin. Now we know that during the gestation period, prolactin levels are very high. And after parturition, when the estrogen progesterone levels drop, it allows for prolactin to secrete milk, to produce milk, right? But the prolactin levels also go down after, parturition. They come to normal levels, but they begin to produce in surges after parturition. It is not like a continuous level. I have a graph here, so it will help you understand. Yeah. So here you see prolactin, which is red, the red line here, and the black and the progesterone, the estrogen and the progesterone levels, right? So, sorry. So after parturition, that means after the mother has delivered the baby, the estrogen and progesterone levels have dropped, right? They've dropped considerably. They were almost zero, right? But prolactin levels also almost come back to normal. But during the nursing time, there are intermittent bursts, okay? There are surges of prolactin. It's not like a continuous phase. That's how it works. That's how prolactin is secreted in the body, okay? They are in surges like this. Okay, so every time the baby, the mother nurses the baby, the nervous signal is sent from the nipples to the hypothalamus, okay, where then allows for secretion of the prolactin from the anterior pituitary gland, which then allows for secretion of milk. Okay, and this surge of prolactin lasts for 1 hour. Okay, the surge of prolactin secretion which lasts for 1 hour. So although the prolactin levels tend to come back to normal, they decrease. There are surges eventually, okay, while the mother is nursing, and every time the mother is nursing the baby, the nervous signal sends a signal to the hypothalamus, which then allows the anterior pituitary gland to then produce prolactin, and that prolactin surge lasts for about 1 hour, during which the secretion of milk happens. Okay, so that's how prolactin functions in the body. Okay, it is not like a continuous surge, or it will not be like a stagnant secretion of prolactin from the anterior pituitary. It will be in surges. This is a good point to remember. So to answer your question, yeah, which hormone stimulates the secretion of milk? It's prolactin. Okay, so the answer is D. Okay, let's move on to the next question. Question number 11. The immediate cause of induction of ovulation in human female is large plasma surge of Saberan 94, A. LH, B. FSH, C. progesterone, D. estradiol. We already discussed this in the menstrual cycle, right? And also previously, it's LH. So answer is A. So luteinizing hormone secretes from the anterior pituitary gland. It acts on the Leydig cells, which secretes testosterone. Okay, that's in males. In females, it allows for ovulation to take place, and it increases 2 days before ovulation and peaking for about 16 hours. It acts on the granulosa and theca cells, converting them to progesterone secreting cells. The rate of estrogen secretion falls 1 day before ovulation, and it also increases rapid growth of follicular cells along with estrogen and FSH. LH causes rapid release of follicular steroid hormone. That's progesterone. I've already discussed this, right? So progesterone allows for proteolytic enzymes to secrete from lysosomes, which then weaken the theca externa cells. Okay, the whole capsule that is formed, it needs to rupture and weaken it. This causes weakening of follicular capsule and swelling of follicles. Also allows for rapid generation of blood vessels into the follicle wall. Okay, so so LH plays a very important role. Okay, it forms the lutein cells, of course. 2 days before ovulation, it takes place 2 days before ovulation. The surge which then eventually helps in forming progesterone, and then which that eventually progesterone eventually helps in ovulation by weakening the swelling of the follicles and weakening of the walls, okay, and also forming new blood vessels in the follicular wall. Okay, so that is LH, and we've already discussed this before, right? So we can move on. After ovulation, it converts granulosa cells and theca cells into lutein cells. This process is called luteinization, and the structure formed is corpus luteum. Okay, let's move on. Yeah, so this, I'm sure you're familiar with this diagram. So you have ovulation over here, and then the corpus luteum forming over here, okay, after ovulation. Okay, so question number 12. Testosterone is produced by, and this appeared in 1993. A. Sertoli cells, B. Leydig cells, C. oxyntic cells, and D. pituitary glands. So testosterone is produced by Leydig cells. Answer is B. Testosterone is produced by Leydig cells, and so yeah, this is a diagram, the interstitial cells over here. Okay, so you have a transverse section of the seminiferous tubules, and you see the Leydig cells over here. Okay, this is pretty direct. Let's move on. Question number 13. ADH or vasopressin is, and this appeared in 91, A. enzyme that hydrolyzes peptides, B. hormone secreted by pituitary that promotes reabsorption of water from glomerular filtrate, C. hormone that promotes glycogenolysis, D. energy-rich compound connected with muscle contraction. So the answer is obviously B. It is a hormone secreted by the pituitary, the posterior pituitary, that promotes reabsorption of water from glomerular filtrate. Okay, so ADH or vasopressin is responsible for that reabsorption of water from the DCT collection tubules and collecting duct. Okay, if you're not familiar with this, you can go back and watch my videos on excretory products and their elimination. I've discussed ADH in much detail, and also the structure of nephron. All right, I can move on. Okay, question number 14. Occurrence of Leydig cells and their secretion is, and this appear in 91, ovary and estrogen, liver and cholesterol, pancreas and glucagon, testes and testosterone. So Leydig cells obviously they occur in the testes and they secrete testosterone. Simple. Answer is D. So Leydig cells occur in the testes and secrete testosterone. Very direct question, direct answer, right? We'll study this more in detail in the reproductive system. Okay, so question number 15. Which gland secretes odorous secretion in mammals? And this appeared in 2000. A. Bartholin's, B. prostate, C. anal gland, and D. liver. So which gland secretes odorous secretion? That's C. anal gland. Okay, answer is C. So anal glands are present in the anal wall, okay, and secrete an odoriferous fluid into the anal canal via anal ducts. Okay, and it is predominant in carnivorous animals, and carnivorous animals they use this secretion that's coming from the anal gland to mark their territory. Okay, the territorial animals, right? So to mark their territory they use that secretion, so there's odor in it, so they know that this is their territory, and they secrete small amounts when they defecate. Now, the glands are often expressed when the animal is anxious or frightened. Also in such cases the gland is expressed. Okay, so you can learn this as a fact that anal gland is a gland which secretes odorous secretion, okay, in mammals. Then move on to the next question. Okay, so question number 16. MSH is secreted by, and this appeared in 2000. A. anterior lobe of pituitary, B. middle lobe of pituitary, C. posterior lobe of pituitary, and D. endostyle. So MSH is secreted by the middle lobe of pituitary. Answer is B. So MSH is secreted by the intermediate lobe of pituitary gland. Now, since intermediate lobe is well developed in lower vertebrates, MSH plays an important role. Okay. The intermediate lobe in the pituitary gland of lower vertebrates is more developed. Okay, and we'll see how that plays a role. How MSH is secreted from there and plays a very important role as compared to humans. Now, fish, reptiles, and amphibians, okay, they change the color of their skin for thermoregulation, camouflage, and behavioral display. So, in nature, fish, reptiles, and amphibians need that. Okay, they need to change the color of their skin. Okay, for thermoregulation, or whether it's camouflaging, or even to show their behavior. How do they do this is that black or brown granules are moved from the pigment cells called melanophores. So there are melanophores called pigment cells. Okay, they are type of pigment cells. They are also called chromatophores, and the black or brown granules present inside are moved. Okay, so here you have a diagram of this is in humans. So you have anterior and posterior, and this is the pars intermedia. So it's a very small structure. Okay, the granules are made up of melanin synthesized from dopamine and dopaquinone. Okay, that's how this melanin are synthesized. The movement of these granules are controlled by MSH, melanin concentrating hormone, melatonin, and catecholamines. See, melatonin we also discussed how it affects pigmentation. It's one of the functions, so that's how it is affecting that. Okay, so it helps in the movement of these granules, okay, from the pigment cells. Now, in some lower animals, the pars intermedia of pituitary gland is highly developed, yes, and is controlled by the hypothalamus based on the amount of light that enters. Okay, so based on the amount of light that enters, also the amount of secretion varies. Okay, and it is seen in lower and some of the lower animals. Now we'll see how. So in Arctic animals during winter months, where it is dark most of the time, animals develop light colored fur. Okay, and during summer they develop dark color fur. So the absence of light is not allowing for MSH to secrete, so they have a light colored fur, whereas during the summertime when there is more light present, it allows for MSH to secrete. So this is an important interesting fact, how in some lower animals MSH plays an important role in changing the color of their skin or their fur in the case of Arctic animals. Also in fishes, amphibian, reptiles, also how this MSH plays a role in camouflaging them, in showing their behavior displays, and thermoregulation also. Okay, so this is in animals, in lower animals. Now mammals have melanocytes that contain melanin granules. Okay, so now we are different. Mammals are a little different from, and I'm talking with respect to humans over here, they are different from the other animals that use MSH to a major extent. But here in humans, MSH is produced or secreted in the blood in very, it's negligible, it's not really there, it's not really playing an important role. We have melanocytes present in our, we don't have the chromatophores like in fishes. We have melanocytes present in us which contain melanin granules in our cytoplasm. But when melanocytes are treated with MSH, it causes darkening of skin within 24 hours. So if MSH is administered, okay, it does cause darkening of skin within 24 hours, which proves that yes, MSH does play some role in increasing the pigmentation of the skin. The pigment cells contain pigment in their cytoplasm, this is melanocytes. Okay, and these pigment cells, the cells are star shaped with long branching processes. They're present abundantly in connective tissue of the skin, choroid, and iris of the eye. Okay, so they are predominantly found there. Now, melanocytes contain melanotropin 1 receptors which bind to ACTH. Now, it's interesting to note that ACTH and MSH have similar structures. Okay, similar structures in terms of their biological sequence. Okay, the amino acid sequence, if you look at it, they are similar. Okay, and ACTH does play an important role in our body. So melanocytes have these melanotropin 1 receptors which allow for ACTH to bind because of the similarity with MSH. Okay, so ACTH binds to that. So if there's an abnormality in the secretion of ACTH in our body, it does affect the pigmentation. Okay, and that we have seen in Addison's disease. Okay, where pigmentation is a issue there. It becomes a abnormality over there. It causes pigmentation disorders, right? Hence, any abnormalities with secretion of ACTH causes pigmentation disorders. Also, congenital synthesis of melanin due to genetic defects can also cause pigmentation disorders. So, in humans, it is ACTH which plays an important role in forming any pigmentation disorders. So, if there is less ACTH or too much of ACTH, you see a change in the pigmentation of the skin. Apart from that, that is a hormonal defect. But if there's a congenital defect, that is at birth, during the sequencing of the melanin during the development of the fetus, then it can also cause pigmentation disorder. So in humans, it is different from animals. Okay, lower animals that we've seen in fishes, reptiles, amphibia, and arctic animals. Okay, in that, because for them, MSH plays a very important role in the way they live. Okay, in nature, whereas for us, we are just born with it based on our genetic makeup. Okay, and also apart from that, ACTH difference or secretion in difference of secretion in ACTH can cause pigmentation disorders. Okay, so that's how we are different from them. Also, studies have found that MSH does not really is produced in humans as compared to animals. Also, we see that the pars intermedia. Where is the diagram for that? Okay, yeah. So pars intermedia is quite small over here, right, as compared to other animals. Okay, so MSH in humans does not really play an important role, but in other animals, yes, it does play an important role. Okay, so here you have a drawing of what these melanocytes look like. So like I said, they're star shaped with long branching process. Yeah, you see the branching process and they look like star shaped. Okay, so this is what MSH does. Now to answer your question, okay, let's go back to the question. Yeah, MSH is secreted by the middle lobe of pituitary. Okay, so yeah, that's a direct question, but I've explained what are the differences, what is the action of MSH, the function of MSH in animals and in humans, how melanin is controlled. Okay, let's move on. Question number 17. The function of oxytocin is to help in, this appeared in 1909, A, childbirth, B, gametogenesis, C, growth, and D, lactation. Okay, so function of oxytocin. Now over here, if you notice, there are two functions. Okay, there's childbirth, yes, and there is lactation also. Okay, so we saw prolactin, we saw the function of prolactin, secretion of milk, but yielding of milk is also a part of lactation, not just secretion of milk, right, from the mammary gland. So the answer is actually A and D. We'll go to the discussion. So oxytocin is produced by the paraventricular nuclei of the hypothalamus and released by the posterior pituitary gland. The functions of oxytocin include Oxytocin secretes in large amounts at the end of gestation, which allow for contraction of the pregnant uterus. So when the fetus is fully developed, oxytocin is secreted in large amounts, which allow for the expulsion of the fetus from the uterus. Another function is that it causes milk to move from the alveoli to the duct so that the baby can obtain it by suckling. Okay, so now prolactin, of course, plays an important role in secretion of milk, but that is all stored in the mammary alveoli. Okay, now it needs to move down the duct and into the nipples where suckling happens, where the mother is able to nurse the baby. A suckling stimulus on the nipple sends a positive feedback to the hypothalamus through sensory nerves. Okay, so every time the baby suckles on the nipples, there's a signal that is sent from the sensory nerve fibers to the hypothalamus, which allows for the production of oxytocin. Now, this causes oxytocin to release into the blood, which causes contraction of myoepithelial cells surrounding the alveoli and allow milk to release. So the ejection of milk is done by oxytocin in the mammary glands. Okay, so that's its job. The secretion of milk is done by the prolactin, but the letting down process is done by the oxytocin. So here you have a diagram of the mammary gland. So you have the mammary alveolus over here. Okay, then you have the mammary duct here, and then the lactiferous duct here, and then finally it goes into the nipple. Okay, so that's how the milk flows. So there are myoepithelial cells. It's not really shown here, but there are myoepithelial cells, okay, which with the help of oxytocin they contract, okay, and they allow for the milk to be let down and to move out into the nipple so that the mother can nurse the baby. Okay, so it also plays an important role in lactation. Okay, in childbirth, yes, of course, it allows for the expulsion of the baby from the uterus, but also in lactation it plays an important role too. Okay. Clear? I guess I've answered this question. The function of oxytocin is to help in childbirth and lactation. Okay, I guess we can move on to the next question. Okay, so question number 18. Secretion of progesterone by corpus luteum is initiated by, okay, A, testosterone, B, thyroxine, C, MSH, and D, LH. And this appeared in 99. I think we already discussed this a million times now. It's D, LH. Okay, let's go to the discussion. Temporary endocrine gland is called corpus luteum. Okay, and after ovulation, the theca interna cells and granular cells form lutein cells, and they enlarge 2 or more times in diameter and become filled with lipid inclusions, giving it a yellowish appearance. Now, this process called luteinization, the mass of cells is called corpus luteum. The granulosa cells develop a smooth endoplasmic reticula that form large amounts of progesterone and estrogen, but mostly progesterone. Okay, so LH helps in the luteinization. They form those lutein cells, and the granulosa cells develop a smooth endoplasmic reticula that form large amounts of progesterone. So the luteal phase is mostly dominated by progesterone. Oh, sorry. Let's go back to the question. So, question number 18, to answer your question, yeah, secretion of progesterone by corpus luteum is initiated by LH. Clear? This was direct. You need to study the menstrual cycle properly to answer this question. Okay, so question number 19. Insulin is it's appeared in 1990, A, vitamin, B, lipid, C, hormone, and D, enzyme. By now, you should know that insulin is a hormone. So, the answer is C. Let's go to the discussion. So, insulin is a hormone that is secreted by the beta cells of the pancreas, right? Now, how is insulin synthesized, right? So, there is a structure called pre-proinsulin, which is synthesized in the ribosomes on the endoplasmic reticulum. So, pre-proinsulin is the first step okay, to the production of insulin, the formation of insulin. Now, once that pre-proinsulin is formed, it is cleaved to form proinsulin. Okay, so from pre-pro, it is formed to proinsulin in the endoplasmic reticulum. So first in the ribosomes, then proinsulin is formed in the endoplasmic reticulum, and it contains 3 chains of peptides A, B, and C. Okay, so it is formed of 3 chains. Now, proinsulin is cleaved in the Golgi apparatus. Now, proinsulin is just the precursor for insulin. So, again, it moves into the Golgi apparatus to form insulin, okay, which is composed of A and B chains connected by disulfide linkages, which is the final structure of insulin. And there is a C chain peptide, okay, it's also called as connecting peptide, if you can remember it that way. Okay, so proinsulin has A, B, and C. Now, proinsulin again gets cleaved into C peptide, that is the connecting peptide, C chain peptide, and A and B, which is the final structure of insulin. Okay, and these A and B chains are connected by disulfide linkages. So insulin and C peptides are packaged in secretory granules and are released in equimolar concentrations. Okay, so you have insulin over here and you have C peptide in the secretory granule. I have a diagram, a structure for that to show the function, the structure of insulin in the secretory granules, and they are released in equimolar concentrations. So normally, Normally 90 to 97% of beta cell is insulin secretion along with equimolar concentration of C peptide. Okay, so yeah, so this is what I wanted to show you. So pre-proinsulin is not here, but proinsulin is here. Now you can see the A chain, the B chain over here, and this is the C chain, and this is the cleavage. Okay, and these are disulfide linkages over here. You can see that A and B chain are connected like that. Okay, the cleavage happens there. So now they are stored in secretory granules. Okay, so you have a C peptide which is separate, and you have the final insulin hormone. You have the final hormone which is then secreted out. Okay, so this is how insulin is formed in the beta cells. Okay, so question number 20. Which hormone possesses anti-insulin effect? And this appeared in 1988. A. Cortisol, B. Calcitonin, C. Oxytocin, and D. Aldosterone. Remember, I have already discussed this in part 2. And see, this question has appeared again. So now if you have watched part 2, you would know the answer to this question very easily. So which hormone possesses the anti-insulin effect? It is A. Cortisol. Okay, so your answer is A. So cortisol is very potent and accounts for 95% of all the glucocorticoid activity. And cortisol promotes gluconeogenesis. How it does it? It increases the liver enzyme activity to convert amino acids to glucose. It causes mobilization of amino acids from the extrahepatic cells, especially muscles. It increases mobilization of fatty acids. It decreases uptake to glucose by cells. An increased rate of gluconeogenesis and decreased uptake of glucose utilization leads to high level of glucose in blood, leading to adrenal diabetes. I've already discussed adrenal diabetes before, but just say that if there is a high amount of glucocorticoid activity, especially of cortisol, it will obviously increase the rate of gluconeogenesis. At the same time, it decreases the uptake of glucose in the cells, right? So that means there is too much amount of glucose present in the blood, and that leads to adrenal diabetes. Now what happens is that increased levels of cortisol result in decreased insulin sensitivity, especially in muscles and adipose cells, hence lowering the action of insulin. So now let's say there is high amount of glucose present in the blood due to gluconeogenesis, but there is decreased uptake of glucose by the cells due to presence of high amount of cortisol, right? So now pancreas notices that there's too much glucose. It wants to get rid of it. That's its job, right? So it begins to secrete insulin. okay, begins to secrete insulin. And the first job is to send it to the muscle cells. Okay, we'll drop it off to the muscle cells and the adipose cells. Okay, we'll drop it off there. But now the receptors present in the muscle and adipose cells are not very sensitive to insulin. They're like, no, we don't want you. Okay, so they are not very receptive to the insulin bringing in glucose there. Okay, now this is why this happens is because due to high amount of cortisol. Now cortisol decreases the insulin sensitivity, it increases the resistance. Okay, it does not. Now cortisol is acting the villain as the villain over here in this story. Cortisol is not allowing for insulin to do its job by decreasing the sensitivity of the insulin receptors. It's like, no, I don't want you. Okay, cortisol is not allowing for that glucose to move in. It's not allowing for insulin to do its job. So that's how it results in adrenal diabetes. So that's how it has what? Anti-insulin effect. Okay, so there is too much of cortisol, there's a high amount of cortisol activity that's happening in the blood, then it has an anti-insulin effect. So yes, that pretty much answers our question, right? So which hormone possesses anti-insulin effect? Yes, of course, cortisol. It's not calcitonin, oxytocin, or aldosterone. So that answers our question. We'll move to the next one. Okay, so question number 21. MSH of pars intermedia middle pituitary is responsible for, and this appeared in 1988. Pretty old question. A. Darkening of skin in lower vertebrates. B. Light coloration of skin in lower vertebrates. C. Both A and B. D. Darkening of skin in human beings. So MSH of pars intermedia middle pituitary is responsible for darkening of skin in lower vertebrates. I have already discussed this. See, if you paid attention, you would know the answer to this question, right? The answer is A. And so it does not really play a role in human beings, but yes, in lower vertebrates, it helps in darkening of skin. So MSH is secreted by the intermediate lobe of pituitary gland. It's the same answer. It is well developed in lower vertebrates. Fish, reptiles, and amphibians take advantage of the situation and they change the color of their skin for thermoregulation, camouflage, and behavioral displays. Okay, so black and brown granules are moved from the pigment cells called melanophores, also known as chromatophores. This is the structure of the pars intermedia. The granules are made up of melanin synthesized from dopamine and dopapinone. The movement of these granules are controlled by MSH, melanin concentrating hormone, melatonin, and catecholamines. And some lower animals, since pars intermedia is highly developed, is controlled by the hypothalamus based on the amount of light that enters. Okay, so now this is where it plays an important role in changing the skin color. So in Arctic animals during winter months where it is dark mostly, animals develop light colored fur. So MSH does not do its job of darkening the color of the fur. But during the summer they develop dark color fur. Okay, so it is controlled by the amount of light that's entering. Okay, so this is how to answer your question. Darkening of skin in lower vertebrates. Yeah, that's the appropriate answer. It's answer A. Okay. So mammals have melanocytes that contain melanin granules, and when treated with MSH causes darkening of skin within 24 hours. Okay, so we've come to the end of this video lecture. And I hope you've enjoyed today's video lecture. I hope you've learned something new today. So don't forget to like, share, and subscribe to MJ NEET Biology. And I'm also available on Facebook, Twitter, and Instagram. So you can follow me there for more updates. And I'll see you in the next video lecture, alright? So bye.